up for this.
All right, well, I guess we should maybe get started. Nobody else seems to be coming in and to the meeting, and it's 3 after. So first off, let me thank everybody for making time out of their day. It's a bit unfortunate we have to do this over Zoom. I wish we could be meeting face to face, but it's just reality of our lives now. So it gives me great pleasure to introduce Zachary Boswell. So this is Zach's defense of his PhD dissertation. Zach joined my group in the summer. He had a special deal to start in a little bit early, so he joined in the summer of 2015. He did his before that he did his bachelor's degree at Hardin-Simmons University, which is in Abilene, Texas. And Zach's actually a native of Lubbock. He was born in Lubbock, and then his parents' family moved to Abilene, and now he's back. So he did his BS at Hardin-Simmons, and then, as I said, joined my group in 2015 as a graduate student at Texas Tech. Zach's actually my first graduate student, and will be the first from the Latham labs. A very exciting day for us. First in the Latham lab to get his PhD. Hopefully, if all goes well.
Yeah, don't jinx me yet.
Yeah, so over the last 4 years, Zach, basically, when we started, Marella, my wife and I, basically had some plasmids. We had done some very preliminary expression tests, and we were like, well, this one sort of works better than the other. We might as well give it to Zach and let him go for it. And he did, and really took off. And the quality of the work shows. He's published 3 papers, 2 first author papers, 2 research, 1 review in biochemistry. Maybe there could be another one out, hopefully before he leaves. We'll see if we can get enough stuff done in a short amount of time. He's, given numerous poster presentations, and short talk, and promoted talks at various conferences. He's won several awards from doing those presentations. He's won a number of awards within the Department of Chemistry and Biochemistry in his time here. So, yeah, it's a bit of a bittersweet day for me in the lab, I would say. And I mean, yeah, here we go. So I would just turn the Zoom over to Zach, and then I guess I'll say, it seems like everybody else is muted, I'm going to mute myself. So if you aren't muted, please do so. And yeah, that way we won't interrupt Zach. And okay.
Great. Thanks, Mike, for the great introduction. You're over-embellishing a little bit, but I appreciate it. So welcome, everyone. Good morning. Welcome to my defense. Today, we're going to be discussing key allosteric mechanisms within the ABC ATPase core of RAD50 and how this plays a critical role in DNA repair. So every day, your DNA is under attack by internal and external stresses. This can include UV radiation or oxidative species that are generated just from cells living. And one particularly bad type of DNA damage are these DNA double-stranded breaks, where the phosphate backbone of the DNA is cleaved on both ends, and then this is no longer attached. It's not straightforward to repair. Requires repair pathways to repair this, but this could be potentially fatal for the cell because the gene can dissociate and no longer be able to be repaired. And this could lead to cell death or cancer. Now, the protein complex that is involved in initiating repair on this specific type of break is MR11 RAD50 MBS1 protein complex. So my project primarily focused on RAD50. So here you can see on the left, this is a cartoon of the sequence of RAD50 nucleotide binding domain. On the right is the crystal structure of RAD50. So RAD50 is an ABC ATPase, which is one of the largest protein families out there. So I have shown here the universally conserved motifs in ABC ATPases. So notably, you can see the Walker B domain and the signature motif. These are involved in ATP binding. The Q loop and the Walker B motif are involved with ATP hydrolysis. And then I want to point out the D loop and the hinge region of RAD50. We will be discussing these throughout this presentation. So this is RAD50 MBD, where it has a significant portion of the coiled domain, that is this part of the structure, removed and replaced with a flexible linker. This is a commonly used construct for structural studies because this smaller sized protein is easier for structural studies. However, in many of our in vitro assays, we will use this full-length RAD50 throughout this presentation. And this involves extra cold coil and the zinc hook domain. So here you can see two crystal structures of MR complex, where in blue are MR11 subunits, so the MR11 will homodimerize. So here's one MR11 subunit, and here's the other MR11 subunit. And this binds with RAD50, and again, there's two subunits of RAD50. And RAD50 modulates the conformational changes in MR complex, which changes the function of the complex. So RAD50 will bind to ATP, and there will be a conformational change to promote this closed state. And this can be reversed by RAD50 ATP hydrolysis. Now, notably, the MR11 active site is right here. And when you enter the closed state of the complex, the MR11 active site is occluded, where DNA can no longer interact with MR11, and MR11 cannot cleave the DNA. So there are still many questions in the MR field, MR11 RAD50 field. Many of these are involved with how MR is moving and how that is related to the function. So remember, in proteins, function is critical for proper activity. If proteins were just rocks floating around in solution, they would not be very interesting. So they can probe conformation states that are functionally active. And NMR is great at detecting these different types of motions at many different timescales. So we use nuclear magnetic resonance spectroscopy as our main tool in the lab. NMR can look at hours timescale motions, which involves like protein folding, protein translocation, all the way up to nanosecond, picosecond timescale motions, which can involve side chain rotations and domain movements. And very notably, we will talk about this a lot throughout the project, nanosecond to picosecond timescale motion is a great indicator of allosteric in proteins. So traditional NMR spectroscopy labels N15 in proteins, and you look at the amide-based NMR spectroscopy of the backbone. However, NMR has this size problem, which I'm sure many of you are familiar with, where the larger your molecule, the worse your spectra becomes because your peaks become too broad. So this is the first ever 2D correlation spectra of proton and N15 that I recorded for RAD50 MBD. Now this is at 600 megahertz, but this is just RAD50 MBD, that is 43 kilodaltons. So each one of these peaks represents one of the amide groups in a backbone. Now this looks pretty decent. We get pretty good differences in our peaks. But we should have approximately 300 peaks here, and we're only seeing about 150. So right off the bat, with RAD50 MBD, because it's too big, we're just straight up missing half of our signals. So we can't deal with that. We want to look at full MR complex, which is a very large complex. So the Latham lab uses methyl-based NMR spectroscopy, which this is C13 labeled isoleucine, leucine, valine, and methionine residues. And these methyl groups have beneficial spectroscopic properties, primarily driven from the methyl groups being at the end of these long side chains in the residues. And at the end of these long side chains, they have faster local dynamics, which this improves the relaxation of the methyl group so that we can have longer lived signal. Now, we also have 3 protons to initiate magnetization compared to the 1 of the backbone. So that straight up gives us 3 times the signal to noise. And then we can also limit the number of atoms that are going to be labeled in the protein, specifically to ILVM residues. So here on the right, you can see the 2D correlation spectra, but now it's in between proton and C13. So each one of these peaks is a methyl group in rad50. We labeled ILVM labeled rad50, and we added in unlabeled MR11. So the MR11 is not showing up in this structure, but it's still contributing to making the full complex. And this thing is 180 kilodaltons. So this is much bigger than the last spectra that I showed you. However, we have very nice peaks, and you can see nearly all of them in solution, where there's very little overlap between the crowded regions. Now you can see that the residue types have these general groups where methionines are over here, isoleucines are up here, valine and leucines are down in this region. Now these have different positions in our spectra because the chemical environment of the residues are different. So say this methyl group is right next to the sulfur group in methionine, so it has a distinct chemical shift, which is different than isoleucine and the others. However, the differences in the peaks within the methionines are from different chemical environments within the protein. So say that this methyl group here is buried in the core of rad50 and it's not very solvent exposed, and so it has this chemical shift. But then say this methyl group that is very different than this one is say in a flexible loop and it's more solvent exposed, then it has a different chemical shift. So each one of these peaks is indicative of the local chemical environment of the methyl group in rad50. So this is all great, except if you don't know which peak is which methyl group in your spectra, in your protein, you don't really know what's going on. And so we had to make peak assignments, and we used this 3D methyl nosy, which will correlate methyl groups through space with one another. So what we do with methyl nosy is we initiate magnetization on the methyl groups, and then we wait. And this magnetization will transfer to other nearby methyl groups if they're close enough. And they will have chemical shift information of that methyl group. So then you can see that these black cross peaks arise in our spectra, and you can make these maps to see which methyl groups connect to each other, and you make all of these connections, and you go back, and we had the benefit of having previously solved crystal structures, so we can go and look at the crystal structure and see which methyl groups make sense. So we can see that this methionine group is next to another methionine, another isoleucine. Hey, this guy is next to a methionine and another isoleucine. Now this is a bit like doing a 3D puzzle without many corner pieces. And so we engineered in some extra corner pieces to help us with assignments. So we made a series of cysteine mutations in rad50, and we used paramagnetic relaxation enhancement to assist with our assignments. So for example, say we made this valine 768 to cysteine mutation. This is nice because we cut out a valine resonance, which will make peaks disappear from the spectra, and we will get 2 assignments based on that. But then on top of that, we can add in a paramagnetic spin label, this nitroxide spin label, and it will disulfide bond with this cysteine. And this spin label has an unpaired electron that will eat up magnetization of methyl groups that are nearby this site. So then we can see here in the spectra, here's 2 examples. So here is just wild type rad50 versus this L51C rad50. And you can see the L51 peak disappears in the mutant. We get that assignment. And then on the right, this is the valine 768C mutation before and after addition of the nitroxide spin label. And you can see that in black here, we see peaks before we added in, and then in red, the peaks disappear. So we know that these peaks are nearby this site, and we can go back to the nosy spectra and try to figure out who they are. Now we did this 6 or 8 times and cover it all throughout rad50, and each one of these is a different mutation, and each one is a different NMR sample. So this is a lot of work to get all of these corner pieces. Now we covered most of rad50, but there was this one pesky region, this hinge region of rad50 that we did not hit with PREs very well. So we just made some straight up mutations from valine to methionine to get specific assignments. So from all of this, We completed our RAD50 peak assignments. We got approximately 90% assigned. The unassigned peaks are in very highly overlapped regions, and it would be hard to be able to tell what's going on there anyways. And this took about 6 to 8 months to get to this point. But we noticed something really interesting happening with our hinge region mutations. Again, this is the 2D correlation spectra between proton and carbon, but now this is the wild type spectra overlaid with our mutations. And you can see that in green here is our wild type, but in orange and purple are our V to M mutations. And they are seeing chemical shift perturbations, which means that the peak is moving compared to wild type, which means that the methyl group has a different chemical environment in these mutations. And so we noticed that these mutations kind of fell on a line. And so this means that there are two states in solution that are being populated at 2 different degrees in fast exchange. So we went back to the crystal structure when we tried to rationalize why this is occurring. And we saw that these mutations are just proximal to this RAD50 basic switch. And this RAD50 basic switch is this arginine 805, and it will form a hydrogen bond, with the backbone of this alpha helix on the N-terminal lobe of RAD50. And this is involved with the conformational change between the open and closed state of RAD50, and where this interaction is present in the open state, but then when you add an ATP and go to the closed state, then this interaction breaks, which should trigger the complex to shift to the closed state. Now we made this mutation, this R805E mutation, which eliminates this interaction, this basic switch interaction, and we saw that it also had chemical shift perturbations that were even bigger than our V to M mutations. And generally speaking, it fell on the same line as our mutations, suggesting that the phenomenon that it is probing is similar to our mutations. So what I want to try to prove to you now is that these mutations are involved in an allosteric network in RAD50 that is both dynamic and correlated to MR11 RAD50 complex activities. So again, we took our chemical shift perturbations for each series of mutations for the methyl group, and we compared each methyl group to one another. So for example, we took this leucine and this isoleucine set of chemical shifts, and we correlated them to one another. You can see that they have a pretty good correlation to one another. We did this with every single methyl group in solution, and we performed some statistical analysis on these to see which correlations were similar to one another. And we got these 3 distinct clusters. And so we can plot these 3 clusters on the crystal structure here. So all of these are methyl groups that are changing upon mutation in RAD50. So again, we're making mutations here in the crystal structure, which you would expect some local changes just from the chemical environment changing, from the residue changing. But what we're actually seeing is long-range structural changes in RAD50 that span all the way over to the Walker-A motif, which this is approximately 50 angstroms away from one another. So this is indicative of an allosteric network in RAD50 that is being altered upon mutation. So our next question was, how are the motions in RAD50 being altered? And we wanted to specifically look at this fast timescale dynamics using a triple quantum forbidden experiment, because this is a great indicator of allosteric changes in proteins. Now, this experiment has both allowed intensities and forbidden intensities that arise. So let me briefly introduce what is occurring here. Don't be scared of this. This is relatively straightforward. So this is the energy level diagram of our 3 protons in our methyl group. So here is the lowest energy state where all 3 protons are in this alpha state. The highest energy state for a proton would be beta here. So we have you can populate these single quantum transitions. These are the allowed intensities that I mentioned. So where one proton would flip to a higher energy state. Now, this is analogous to, say, absorption, to where you're taking the ground state and you're exciting it to an excited state. This is an N=1 transition and is allowed by traditional spectroscopic definitions. However, Since we have 3 protons that are attached to a very large protein, we can play some tricks with the NMR where we can shift 2 of these protons to a higher energy state at the same time, or even all 3 of the protons to the highest energy state at the same time. So when I say triple quantum forbidden transition, that just means that the 3 protons are shifting to this higher energy state all at the same time. And you can see that these intensities grow in over time. So each one of these blocks is at a different delay time, where the forbidden intensity grows in and the allowed intensity will actually relax away a little bit. And you can plot the ratio of these intensities where forbidden is on top and allowed is in the denominator. And you get these buildup curves. And these buildup curves report on the local motion of the methyl group. So to put this in context, let's say we have this methyl group here, and it can move within this cone. And this cone is pretty big. This is a pretty flexible methyl group that can move all over the place. So this makes it to where the forbidden intensity grows in slowly at different delay times. But then let's say there's another methyl group in the structure that's buried. It doesn't have much flexibility, so it can only move within this cone. This changes how the forbidden intensity grows in. So let's say it's pretty rigid like this. So then the forbidden intensity would grow in more rapidly and then plateau. And we can use these buildup curves to compare our dynamics in the methyl groups in RAD50. So these are our buildup curves for wild type and our mutations. You can see some mutations don't change, or some methyl groups do not change very much, and other ones do. And we can plot this again on the crystal structure, where red and orange are becoming more flexible upon mutation, and these primarily lie on the backside of RAD50. And then in blue and green are methyl groups that become more rigid, and this is more in the middle of RAD50. And interestingly, red and green correlated well with our changes in chemical shifts that I reported, and orange and blue did not. So now we see that we have mutations in RAD50 that change the structure and the dynamics of the complex. We want to ask what is happening to RAD50 and MRE11 functions. So we will discuss 3 of these: ATP hydrolysis, RAD50 dimerization, and MRE11 exonuclease. First, we looked at RAD50 ATP hydrolysis. And so a reminder, ATP hydrolysis is where 2 ATPs will bind to the MR molecule. And then over time, this will be converted to 2 ADP molecules, and we will get this free phosphate. So this assay uses a colorimetric receptor that will bind to the free phosphate, and this will induce a green color, a change to green color. We can measure this green color via its absorbance, and we can convert it to moles of phosphate using standard curves. So here you can see the velocity of ATP hydrolysis versus the addition of ATP. And so this is wild type in our mutations again, where we also included this K36M mutation, which is a hydrolysis dead mutant. So you can see it's here at the baseline. We will discuss this guy more later. Now, this doesn't look super impressive, but it's because we had to reduce our protein concentration in our mutants by quite a bit so that we can stay in the linear range of the assay. So when we normalize to the enzymatic concentration and normalize it to the wild type activity, you can see here that wild type is at 1. There's a slight increase in the valine 156M mutation, but then valine 160M and R85E just go nuts, where they have about 8-fold increase in activity versus wild type. Now, this was a bit peculiar to us because, structurally speaking, we're essentially throwing a wrench in the gears of RAD50, and we expected to break the complex. We expected ATP hydrolysis to be reduced. However, we're seeing that throwing a wrench in the gears actually increased its activity. So we went and looked at the dimerization of RAD50. So this is just RAD50 MBD, and we wanted to see how the RAD50 is closing. So we just simply use size exclusion chromatography to look at this, where large molecules will be eluted earlier in the column and small molecules will come off later. And so the idea here is that we look at RAD50 in the absence and presence of ATP, and we can see if a dimer population comes in because it will be twice the size of the monomer. So you can see here in gray is RAD50. In the absence of ATP, we get this nice peak at this monomeric size. We add an ATP and we lose some of this monomeric population that moves into the closed state. And it's very obvious that our mutations don't mimic that of wild type. They actually increase the amount of population that's processing the closed state. Which again, this is interesting because this means that these mutations, these structural changes in RAD50 are allowing RAD50 to enter the closed state more readily. So then our question is, are all of these changes in RAD50 going to translate to changes in MR11? So to do this, we use this 2-aminopurine fluorescently labeled DNA. You can buy these from companies. So for this assay, we tagged the second nucleotide in from the 3' ends. Now, MR11 has 3' to 5' exonuclease activity. So if MR11 comes in and starts chewing up this DNA, then this 2-aminopurine will be released from the DNA duplex. When it's released from the DNA duplex, it becomes fluorescent, and we can measure this. So here you can see that we measured the exonuclease activity in the absence of ATP, and then in the presence of ATP, and then with this AMP-PMP molecule, which is a ATP analog that is non-hydrolyzable. And the non-hydrolyzable analog kills activity across the board, essentially. And then in wild type, you can see that there's no stimulation upon addition of ATP, but then in our mutations, they actually see increases in MR11 exonuclease with ATP. Now, this is a bit backwards because the first thing I told you was that the closed state of RAD50 blocks the MR11 active site. But what we're seeing here is that these mutations that more readily access the closed state is actually increasing MR11 activity. So we took all of these activities and we compared them to our chemical shift perturbations that I mentioned earlier to see which methyl groups correlated to activity. And very interestingly, I love this piece of data. Cluster 3 entirely disappeared. So none of the methyl groups that were in cluster 3 correlated well with activity. Only cluster 1 and cluster 2 correlated well. And so you can see these are the methyl groups that are related to activity. And if we look at this, in the closed state of RAD50, this is a beautiful illustration of the allosteric network in RAD50, where if we poke it here, it changes methyl groups all the way over here to make this nice box that transcends the RAD50 protomers in closed state. And all of these methyl groups are related to activity. So shifting gears here, that was all part of my scientific reports paper. Now this is part of the nucleic acid research paper. But this was motivated by this cancer discovery paper that was published in 2014. And this paper was involved in a clinical trial where they used DNA damaging drugs and a CHEK1 inhibitor to try to kill cancer cells. So these patients overall did not respond well to treatment, but there was one woman that was 100% cured of cancer within 3 months, and then she came back years later and was still in complete remission. So this whole time they were genome-wide sequencing the tumor cells versus the normal cells in all of these patients. So they went back and looked at what was different about this woman to let her survive the cancer. And they found that there was this L to F mutation in RAD50, and they saw that RAD50 is a DNA repair enzyme, and so they proposed that this was causing changes that would block ATM activation, that would shut down the other repair pathway and lead to cancer cell death. They looked at some in vivo assays with yeast. This is just a yeast survivability assay, where they did this L to F mutation. And you can see with DNA damaging drugs that actually L to F survives pretty well. It survives like the wild type. They also looked at this D to N mutation that we will be discussing. This is a breast cancer mutation, and it's dead already from the get-go from DNA damaging drugs. Now they block MEK1, they remove MEK1, and this is essentially like mimicking the CHEK1 inhibitor, and now L to F dies and D to N is still dead. So they see some differences between L to F and D to N. And they said, okay, L to F is killing these cells while wild type is still okay. That's probably how it's involved with these cancer cells dying. And they left it at that. So this is like perfect for the Latham lab. So now the Latham lab comes in and says, okay, what is happening structurally, functionally, and dynamically in this L to F and D to N mutations that are going to play a role in cancer? So it turns out that both of these mutations are right next to each other in the RAD50 structure. They lie in this ABC ATPase universally conserved D loop, shown here right next to the signature motif. And not many studies have been done on the D loop. There have been some observations in crystals where you can see that the D in the D loop, so the L and the D are universally conserved, that the D in the D loop will form a hydrogen bond with the trans RAD50 Walker A motif. And they said that this should stabilize the closed state of RAD50. Now, the L in the D loop, right next door to the D, will fold in upon inducing the closed state. So this will fold back towards the hydrophobic core of RAD50. But then in the open state, this is flipped back out to where it is more loosey-goosey flopping around in the wind. So now I want to prove to you that changes to the RAD50D loop results in a less stable closed state, but actually increases the ATP hydrolysis rates. And then that L to F probe's a unique state that alters RAD50 allosteric. So now our first thing that we looked at is RAD50 ATP hydrolysis, but we got a little bit fancier with this assay this time. Now we're using the full length RAD50, and we're doing this in biomol green again that I showed earlier. So now we want to also know how RAD50 ATP hydrolysis is related to a DNA detection. So we took the wild type and we looked at it before and after addition of DNA. So you can see that without DNA, RAD50 is really bad at hydrolyzing ATP. It is has very low velocities. But then we add in DNA and the activity goes wild. And so we get a whole lot of stimulation upon addition of DNA. We did this with the mutations as well. So this is the L to F mutation, and we get an increase in RAD50 in ATP hydrolysis in the absence of DNA, which this is similar to the previous mutations, the hinge region mutations that I showed you. But then we add in DNA, and there's very little stimulation compared to the wild type. We look at D to N as well. And again, there's another increase in ATPase stimulation versus the wild type, but again, an even smaller stimulation upon addition of DNA. So you can see this relative change here in these bar charts. And notably, the Hill coefficients of this Michaelis-Menten curves are all greater than 1, which suggests that there is some allosteric activation that could be involved with the DNA stimulation in this assay. So we wanted to ask what is happening in the closed state of RAD50 again with these mutations. So we got fancier, we evolved as we grew in the lab, and we started using this luminescence resonance energy transfer, or LRET, to look at the closed state. And this is very similar to FRET, which many of you will be familiar with, but we add in a cysteine mutation. So this is the L51C cysteine mutation that I discussed earlier. And we can add in some fluorescent tags. So this is a terbium tag where we can excite this terbium. And if it's far away from BODIPI, then it will have some emission that we can read, but BODIPI will not be excited. But now, say RAD50 probes the closed state, these dyes will come closer together. And when we excite the terbium, then energy will be transferred to the BODIPI molecule, and we'll actually be able to detect the acceptor emission. And the efficiency of this transfer from terbium to BODIPI can be used to calculate the distances between these two molecules. So what you see here is the exponential decay of BODIPI signal over time. So this is the acceptor signal that is stimulated from terbium excitation. And we look at this before and after addition of ATP, and you can see we get this nice increase from ATP, which is not surprising because we should be getting more closed state and wild type, so that means that these molecules are closer together. Now we do this in L to F and D to N, and the intensities across the board are lower to begin with, and ATP does not induce much stimulation into the closed state. So what we can do is we can take these exponential decays and fit them to multiple exponential equations, and then you can get these distribution plots, which these distribution plots show the populations that are in solution that are contributing to these decays. And interestingly, we see more than one state in solution. We expected just to see this closed state. So this closed state at about 35 angstroms is perfect. This is what we calculated from the crystal structure for the distances between terbium and BODIPI. But we're also seeing this partially open state, which this isn't fully open, because if it was fully open, then it would be invisible to this LRET pairing, because the molecules are too far away from one another. Instead, what we're seeing is something that's at approximately 45 angstroms, which might look something like this cartoon. So we look at this in L to F and D to N again, and for the closed state, the true closed state, we're not seeing many changes upon addition of ATP. And we're also detecting this partially open state in these mutations, and there are some changes upon addition of ATP, but we're not exactly sure what's going on in this partially open state yet. So to confirm that these mutations are not populating the closed state very actively, we went back to old reliable, to the site exclusion chromatography. So again, in green, and wild type is here. So this population is the closed state of RAD50. And you can see that the mutations do not populate the closed state very stably at all, maybe 2 to 5% shift to the closed state. So this so far is backwards from what I showed you earlier. So right now we've seen increased hydrolysis rates, but decreased RAD50 closed state, which last time we saw both increases in hydrolysis and RAD50 dimerization. So we asked, how can this be happening? Now, we used LRET again, which this is a very cool technique in LRET. You can actually add an ATP and watch the complex change over time. So at the beginning here, you can see that there is low BODIPI intensity, and then the intensity increases over time. Which this again means that there is more closed state in solution. Now for wild type, this takes up to 10 minutes to actually get to fully closed for this to equilibrate. But then we look at L to F and D to N in this method, and you can see that there are lower amounts of closed state again, which is not surprising, but they equilibrate really rapidly. And this equilibration is the sum of the on and off rates for this complex, this transition here, which is this increase in equilibration is probably primarily driven by an increase in RAD50 dissociation because we are not seeing increases in RAD50 dimer. And so we can plot these relative equilibration rates versus one another, and we compare them to our hydrolysis rates that we collected, and you can see that there's a very good correlation between RAD50 equilibration and ATP hydrolysis. So finally, we asked what's happening with MR11 again. And again, we got fancier with our assay, where we moved in this 2-aminopurine tag, 11 base pairs into the construct now instead of being here on the end. So if we are getting exonuclease activity, it means that RAD50 is being, or MR is being efficient in chewing all the way into this instead of just sticking on the end. Now to get this activity, we need to use full-length RAD50, and ATP is required for this processivity. So you can see here in the absence of ATP, we get very little signal, no signal. We add in ATP and we get a large amount of exonuclease activity, which means that the complex is chewing up all the way past this molecule. And with AMP and PMP, the hydrolysis dead mutation or analog, you see that activity is killed again. And then in our mutations, we have absolutely none of this exonuclease activity, which this is not surprising. If this activity requires the closed state of RAD50 and these don't have the closed state, then it's not able to chew up DNA all the way deep into this DNA. So far I've told you that both of these mutations have increased hydrolysis rates. They both have less ATP stimulation, ATP hydrolysis stimulation upon addition of DNA. Their closed states are less stable. They equilibrate faster than wild type, and there is no ATP-dependent MR11 exonuclease activity. So far their activities are very similar. So our question was, what are the structures and dynamics of these D loops also similar? It would be logical to think yes. So far, everything has been the same. But actually, the answer is no, absolutely no. So here you see the NMR 2D correlation spectra between proton and carbon again. This is ILVM labeled RAD50, wild type versus the L to F and D to N mutations. And you can see that L to F in the orange spectra has these nice chemical shift perturbations, and D to N has barely any changes. We can plot these on the crystal structure again, and you can see that here the site of mutation for L to F causes these large chemical shift perturbations that span throughout RAD50. Again, indicative that we're having long range communication between this site and all the way up to the P loop of RAD50, which is indicative of allostery. But then in the D to N mutation, we see very minor changes, and most of them are in this trans RAD50 dimer interface. So we looked at the triple quantum forbidden experiment again to look at the local motions of the methyl groups. And again, we see large changes in L to F and minor changes in D to N, where L to F is generally speaking becoming more flexible. So what we think is going on, D to N is actually a pretty straightforward explanation. So changing this D to the N residue will remove the ability for this residue to form a hydrogen bond with the trans RAD50 interface. And if you remove this interaction, then all of a sudden this the closed state becomes less stable because you're removing one of the key interactions that stabilizes this, and they dissociate faster. But we think that L to F is actually unable to properly fold because now you have this bulky aromatic group that's here. So maybe the loop is not able to fold in the closed state, and we're getting these changes even before in the open state. So maybe the D loop is improperly folded where this hydrogen bond isn't oriented properly to interact with the trans RAD50. So we're getting these intermediate changes in activity and RAD50 association. So this is a model of what's happening so far. These top two cartoons are essentially our first story with the hinge region mutations, where RAD50 will bind ATP and then will populate this inactive closed state. Where the hinge region mutations would have increased this top arrow where it would more readily process, more readily populate this closed state. But then in this closed state, something has to happen for RAD50 ATP hydrolysis to occur. Now, this is probably allosterically activated from DNA sensing, and it's regulated in the absence of DNA. So let's say that a DNA molecule binds, it activates RAD50 ATP hydrolysis by populating this activated closed state, this active closed state, which will do ATP hydrolysis and dissociate from one another and be able to re-enter the cycle. So this is the wild type. What we think is happening for our D loop mutations is that this state, inactive closed state, is no longer present, and instead we get this inactive closed state that is unstable because we're messing with the hydrogen bond that interacts with the trans RAD50. So this is no longer stable. It will actively move into this ATP hydrolysis competent state and then be able to move through this process really rapidly. Now, this would show up in our activity assays as increased ATP hydrolysis, but then also would have less closed state because this guy is no longer present or is no longer heavily populated. And then the DNA stimulation that gets around this allosteric regulation would no longer have a big effect on the complex, which that agrees very well with all of our functional data that we showed. This also agrees super well with the model that the paper in 2014 proposed. So they said that the L-to-F mutation was doing something to this closed state of RAD50 that would not be able to sense the DNA damage, which would not be able to activate ATM, which would block this repair pathway. We now know that this closed state of RAD50 is no longer stable upon this mutation, which rationalizes why ATM is not activated. So the very obvious question is, okay, so this woman survived her cancer from having this mutation. Can we mimic this L to F effect in wild type RAD50? And to do that, we tried to develop some small molecules to mimic the L to F state that would bind to wild type RAD50. And then we could use this with DNA damaging drugs to try to kill cancer cells. So we used Autodoc Vina to try to do this in the computer. And essentially you give Vina a crystal structure, you tell it an area that you want your small molecules to bind. You can assign some flexible residues so that these guys can move around in solution and mimic more what is happening in the cell. And then you get out these drugs that will bind to RAD50 that have these respective scores. The lower the score, the better the binder. These are 4 candidates that we bought that targeted the hydrophobic cavity of RAD50. And so we wanted to test if these stimulated RAD50 ATPase activity to mimic that of L to F. So L to F had increased hydrolysis rates. First thing we want to test is, do these drugs also increase the hydrolysis of wild type RAD50? And unfortunately, the answer is absolutely no again. So what you're seeing here is each point is essentially the Vmax of the max velocity of ATP hydrolysis. And so we would expect that if the drug was mimicking L to F, that increasing the drug concentration would also increase the ATP hydrolysis rates. But even up to 100 micromolar of the drug, we are not seeing any effect. So we got unlucky there. But another target that we tried to bind is the D loop specifically. So the idea here is that we would bind the D loop that would alter the D loop folding and try to mimic that of L to F. And so we ordered all of these drugs, which we ordered 8 this time instead of 4. So we doubled down a bit. But unfortunately, these were delivered the same day that the lab was shut down for this little thing called coronavirus. So finally, I want to take the last little bit. So let me check my time here. Okay, I'm good on time. I want to take the last little bit to talk about DNA binding to RAD50 in the closed state in MR complex. So this is probably one of the muddiest topics in the MR literature. We know a lot about it, but none of the data really jives well together. So far we know that a DNA has to interact with RAD50 in the closed state to have telomere maintenance. However, we also know that the MR11 active site must be accessible for nuclease activity, which means that the closed state actually has to shift in some way to allow the MR11 active site to be able to bind to DNA. We also know that MR is required for initial in-processing and DNA double-stranded breaks, but often these DNA double-stranded breaks are bound with, this fly is trying to get to me, are bound with end-blocking proteins like Ku, but it is shown that RAD50 can actually cleave off this Ku protein. So all of this is confusing separately, and so we want to try to bring this together and try to rationalize how all of this could be related. There are a few crystal structures of RAD50 in the closed state bound to bound to DNA. So this is a short DNA construct that binds to the RAD50 P loop in an asymmetric way. This is another crystal structure of MR complex now in the closed state, and a longer DNA construct. And this binds beautifully right in the middle of the groove of RAD50, but there's a lot of density here that's not shown in the crystals that hangs off, and there's no sign of the DNA able to interact with the MR11 active site. So we want to take Rad50 in the closed state and look at it in the NMR while it combines DNA. So the first hurdle that we had to get through to look at this is to eliminate Rad50 ATP hydrolysis. Because NMR experiments take a long time and they're at high temperature, up to weeks, then all of the ATP in the sample will be chewed up into ADP over time, and we will inevitably end up with everything in this open state. So the first thing we did was we made this K36M mutation, which removes the universally conserved Walker A lysine residue. We showed you earlier that this has no ATP hydrolysis, and there are minimal changes in ATP binding affinity. So this guy is a good candidate to try to mimic, be used as a wild type mimic in the NMR while having ATP hydrolysis deficiency. But our question was, does the structure of Rad50 change just from this mutation? Answer is no. So you can see here in the black and the purple spectra that they overlay quite well. So we can use this mutation to try to look at DNA binding. But first, we have to populate the closed state. So we added in ATP to this K36M MR sample. And you see we get these beautiful chemical perturbations to a distinct state from this APO ATP-free state to this ATP-bound closed state. Now, there's a lot going on here, especially in this blob. It's very hard to tell what's going on throughout Rad50 because we don't quite have assignments for all of these shifted peaks here in the blob, but we can use these isolated residues to be used as markers to try to figure out what's going on in Rad50 on a larger scale. So now we've made the closed state in the NMR with this K36M mutation. Let's add in some DNA. So we added in 5:1 excess double-stranded DNA. And if it jived well with the crystal structure that I showed you, and it just bound on top of the Rad50, then we would expect the methyl groups that are near this DNA binding to see some additional chemical shift perturbations that would show DNA binding. However, what we saw were not additional chemical shift perturbations, but instead we saw populations of the ATP-free open state come back. And this was very peculiar for us because this suggests that there's a state in solution that's approximately 40%, 35% populated compared to the closed state that looks like the ATP free state. So now the question is, does DNA knock off ATP? Now, this hasn't been proposed in the literature, especially in the absence of ATP hydrolysis. And so we had to kind of develop a new assay to try to look at this. And we used fluorescent polarization of a fluorescently labeled ATP molecule. And the basis of looking at polarization is that you can detect the binding of this ATP because without any protein, this ATP molecule is moving around very rapidly in solution. We add in MR complex that's moving very slowly in solution because it's big. And then when this ATP binds, then now this molecule becomes polarized because it's tumbling much slower. And you can see this polarization here marked at about 250 polarization value. But we want to look at the closed state, and so we have to add in some unlabeled ATP, and you want to add in enough unlabeled ATP to where the other Rad50 binding site is going to be bound so that it can enter the closed state. You want to be a little bit careful here because if you add in just way too much ATP, then you will actually directly compete off the fluorescent ATP and you will lose all of your polarization. So you can see here we titrated up to 150 micromolar of unlabeled ATP. And we do get some fluorescent ATP knocked off, but not all of it. It would take approximately 1 to 10 millimolar ATP to completely knock off this fluorescent ATP. So we have about 50 polarization to work with on these endpoints. Now in this 150 micromolar ATP point, we have approximately 90% of the closed state. So now we have closed state with some fluorescent ATP bound. Let's add in DNA and see what happens. Does this fluorescent ATP actually get knocked off? And so we look at our sample that has 0 micromolar cold ATP, and you can see that the signal does not change over time. So the fluorescent tag is not getting competed off in the open state. However, in our samples that have more closed state, we actually see that the fluorescent ATP is getting knocked off. And approximately 40% of the ATP molecules are being removed from MR, which this is eerily similar to our NMR data where we saw about 35 to 40% returning to this ATP-free state. So this is pretty strong evidence that ATP is being knocked off by the DNA molecule. Excuse me. So we tried to come up with a model to try to rationalize what's going on here. This is a pretty simple, straightforward model to where we have two stable states in solution, where one state has ATP or DNA bound to the Rad50 dimer on top, just like the crystal structure I showed you. But this guy doesn't have any chemical shift perturbations, and it's about 60% populated. So it just looks like the closed state of the MR complex. However, our data suggests that there could be this state that causes ATP to unbind, where presumably DNA would be binding both Rad50 molecules that looks like it's in the open state because it's dissociated, and also interact with the Rad50 dimer and the MR11 active site. Now, I don't really like this model. This goes against my gut feeling because over time, energetically speaking, this should be a more stable interaction where because the DNA can interact with both Rad50 molecules and it can interact with the MR11 dimer. So eventually over time, I would expect all of this population to shift to this open-like state. However, up to weeks of observing our spectra at high temperature, we still see two populations, no matter how long we wait, pretty much. So we tried to rationalize how we could get two populations that arise from MR in one molecule. So this is the top view of MR complex to where we think that one molecule of ATP could be unbinding, and that would promote two halves of each Rad50 protomer to be mimicking that of the open state. So then this oval part of Rad50 would be unbound ATP, and this would allow the access to the MR11 active site where DNA can come in and bind to MR11. But the other side of Rad50 is still bound to ATP, and where Rad50 can be bound to the closed state of Rad50, DNA can be bound to the closed state of Rad50, where Rad50 is acting like a guide into the MR11 active site. Now, we haven't had much time to try to see direct evidence of this. I set up some crystallography screens to try to see if we can isolate this guy in K36M, but I haven't been able to check yet. But there is some precedent for this in the literature. This is a review by Dr. Altenberg at TTUHSC, and this is an ABC ATPase family member, but this is an ATP transporter, ABC ATPase transporter, to where there are these two models where the ABC ATPase can fully dissociate and allow for ATP to be turn over. But there is also this constant contact model where only one subunit of only one site of ATP binding is active, where that cleaves into ADP. And so this other site would never hydrolyze ATP and would always be in contact. So in conclusion, I hope that I've shown you that Rad50 has an allosteric network that is dynamic and correlated to MR11 Rad50 complex activities. We saw that changes in the Rad50 D loop results in less closed state, but actually higher ATP hydrolysis rates, and that L to F probes a unique state that alters Rad50 allosteric. We tried to mimic this L to F state using small molecules that we developed in silico. And then finally, that DNA forces this ATP-bound closed state into a partially open complex in the NMR. Wow, so I'm to the acknowledgement slide. I want to acknowledge all of my amazing lab members. Many of these wonderful people are on papers with me. Samir did the exonuclease data, the very first exonuclease data that I showed you in Rad50 MBD, within Rad50 MBD. Lily works on many projects, and she is a powerhouse in the lab. Mahtab works primarily, and Henry worked primarily on MR11 in the lab. Tanner is one of our newer graduate students with Henry, and he's working on the MBS1 protein that I did not talk about. But he did a few of the assays in our nucleic acids research paper. Unfortunately, the data just wasn't quite sexy enough to make it into this presentation. Sorry, Tanner. And then Stephen is an undergrad that was in our lab that just loved us too much that he refused to go away. So now he's a research assistant. And then Julie is actually a Welch high school student that visited our lab. There's just no words to describe Mike and Marilla and how much they mean to all of us and how much they contribute to our research and our developments as scientists and as students. So instead, I'm going to tell a story about Julie. I'm not a baby, I'm not going to cry. So Julie is a high school student, right? So she came and she visited our lab for 6 weeks over the summer. And she helped me start to do the LRET studies in the RAD50. And you saw some of her data today. So she's one of these knockout high school students that has perfect GPA, perfect SAT scores. She has full rides to any MD PhD program that she wants in Texas. Now, she chose to come to Texas Tech because of how much she loved the Latham lab. She refused offers from Rice, from I'm pretty sure UT, and she chose to come to Texas Tech because of Mike Latham and Marilla Canning. That shows you how, yeah, y'all get the picture. I would like to thank my beautiful photogenic committee members, Dr. Ben Wiley and Dr. Weber, here on the left and right, for sticking with me for 5 years, having to listen to this presentation iterated 5 million different times. I would like to thank Dr. Sutton on the left, or Brian. And I tried to do a lot of protein crystallography in his lab. None of it really ever worked. So I think it's a Brian problem. It's definitely not a Zach problem. I joke. And then on the right is Dr. Altenberg, which he allowed us and taught us how to do LRET, all of our LRET studies that showed us more about the closed-state and more complex. I'd also like to thank our dean's representative today that graciously accepted to listen to this talk and read my dissertation, Dr. Gallahan in the Department of Biology. And then I would like to thank our funders, NIH, CPRIT, and the Welch Foundation. Specifically, the Welch Foundation paid for my salary for my time here. Okay, and with that, I would like to take any questions. And I found a great Calvin and Hobbes cartoon that actually said Spectra in it, so I couldn't not include it.
Okay, well, so join me in thanking Zach virtually for a great presentation, and I guess we'll open the floor to questions from people, so not on his committee. The committee is going to have ample opportunity to ask questions. So yeah, anybody in the general audience who has a question, feel free to unmute yourself and ask away.
I love questions, so hit me with it.
Okay, this is Guillermo Altenberg. I'll ask you a question.
Hi.
Hello, how are you? I have, this is, the protein is from a thermophile, correct?
That's correct, yes. I did not mention that in this presentation.
Are all the different kinds of experiments, say for example, ATPase activity, LRET, NMR, done at the same temperature, and how close it is to the optimal temperature?
That's a great question. ATP hydrolysis is done at 65 degrees Celsius, which is the optimal temperature for hydrolysis in P. furiosus. LRET is done at 50 degrees, and NMR is done at 50 degrees. And that it's not all the way up to 65, and that's mostly on the hardware side. So as you know, the fluorometers can heat up to 50 degrees, but can't go much higher than that. So we got it as close as possible as we could to the optimal temperature. The NMR we can heat up in Lubbock all the way up to 80 degrees Celsius, and it's okay for a long time. But our assignments were done at 50 degrees because we used the 900 megahertz magnet in Denver, and that can only heat up. They'll only allow that to heat up to 50 degrees so that they don't accidentally hurt their $5 million magnet.
Okay.
But
No,
the activity assays are absolutely at high temperature.
And not exactly related to that, but when you did the dynamics of the methyl groups,
mm-hmm.
and those dynamics are much faster than the activity of the protein.
Yes, that is correct. These are reporting on the side chain motions.
Like more than 1 million times faster. Approximately, because you have
I don't know if 1 million is correct. I would say at least 3 orders of magnitude.
Okay, well, let's say it's okay. So what is the functional importance of those dynamics then?
Right. So this timescale dynamics is a great indicator in allostery. So here's a different overlay of different motions in proteins, and so you can see here between the nanosecond and picosecond timescale dynamics is a great spot where allostery can be measured. And we were seeing how the allostery in RAD50 was highly correlated with our activities. And so this is these motions are probably related with the transition between states in the NMR that are related to allostery.
Oh, sorry. Maybe that I don't see this. I don't looking at this the right way. But the equilibration when you do the dynamic took place in about 20 milliseconds, more or less 20, 30, something like that, which will be occurring basically events like 50 times per second. But your hydrolysis is much, much slower than that.
That is true. That is absolutely true.
So
yes.
you have a dynamic that is correlated, but on a completely different time frame from the activity.
That is correct, yes.
So that means that there have to be many of those allosteric changes, and most of them do not have a functional correlate, and only once in a while, very rarely, hydrolysis.
So our changes in dynamics, I wish I was in person to show this, but so if the methyl group is actively processing this, then one of these states is probably related to activity, right? So if it is able to process this faster and faster, then maybe it is able to populate the state that is functionally relevant more rapidly, which would translate into more rapid hydrolysis. But I agree, it is a bit confusing that we're measuring a much faster timescale dynamics versus our hydrolysis rates, but we're still seeing correlation.
Okay, thank you.
Great question. I appreciate it.
Well, if nobody asks any other question, I ask. But I don't want to.
No, I think go for it.
Okay, so I have, when you look, well, let's say the wild type, but it can be with any of the mutants also. When you do the chromatography, the size exclusion, the SEC chromatography, have you ever done this, or are any of your data with ATP Without
Yes.
Basically,
Yes.
no hydrolysis, but ATP bound.
That is correct. And so we've done this with ATP gamma S and AMP PMP, and both of them produce this closed state, yes. But I will say that even with ATP present, this is done at 4 degrees Celsius in the column, and it takes the column, 20 minutes or so for this to elute. So there isn't much hydrolysis that's going on at 4 degrees with our thermophiles, even with ATP.
Okay. And so the fractions, let's say in the wild type, that do not become dimers at the saturating ATP concentration, are those inactive molecules or do you have?
I don't think they're inactive. I think that we just have this dynamic equilibria between the open and closed state in RAD50 MBD. Now remember that there is no MRE11 in this. We wouldn't be able to see a size change between open and closed state if MRE11 was present, because it would all be part of one molecule. So only a small portion, 40% or so, is converting to a stable closed state. This is probably able to process it, but maybe it doesn't get there in time, or it could dissociate before it's able to come off the column.
So basically, just to see how it works. So this will basically bind ATP, form dimers, and dissociates without hydrolysis.
That's correct.
Okay, thank you. Okay, since nobody asked a question, I'm not going to ask a question. I just want to say a really good presentation and very nice work.
Thank you. I appreciate that a lot.
Yeah, congratulations.
Okay, well, do we have any other questions? Going once?
Blah, 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 blah.
Okay, well, I guess if we don't have any other questions, join me again in thanking Zach. And then if you're not part of the committee, it's time for you all to say goodbye. And the committee members will just stay on this chat and the Zoom meeting and ask Zach questions.
Thank you all for being here.
For being here. Yeah, we appreciate it.
Okay, bye.
Bye.
Good luck.
Thanks for the great questions.
Good luck, Zach.
Thank you.
Bye, Zach.
Bye, Derek.
Now everyone say bye.
Bye. Good luck.
Thank you.
Bye.
I appreciate
Bye,
all
Zach.
the.
Great work.
Thank you.
Got it.
Do we clap now, Zach?
No, it's okay. I can hear it in my head.
Okay. Best of luck, man.
Thank you.
Goodbye.
Let's see. Can y'all can still see my screen, correct?
Yeah.
Mhm.
Okay.
Okay, let me. Okay, I guess that's seems like we're now just us. So, I guess now, let me stop recording. I guess, yeah, we stopped recording with Nazmul and Neil, right? And this is not part of the public